It's February 28th, 1953. It's a gray winter day in Cambridge, England, and in the Eagle, it's lunchtime. The pub is dim and cozy, and the smell of the local favorites like shepherd's pie and bangers and mash fills the air. It looks set to be a perfectly ordinary afternoon. But a moment later, the calm is shattered. The front door flies open, and everyone turns to see a blond man in his mid-30s hurry in. On a wooden bench on the other side of the pub, a skinny young man with wild hair and big eyes is sitting. The blond man orders the usual. 2 pints, please. The bartender slides his beer across the bar. Then the blond man raises his glass and calls out for everyone to hear. Gentlemen, we have discovered the secret of life. Around the pub, the reaction is one of shock and disbelief. Some of the other scientists look confused, others amazed, a few look skeptical. What are you going on about? The secret of life? Yes, what is he talking about? The blond man is Francis Crick, an English scientist, and his wild-haired friend is an American, James Watson. The reason for their excitement is standing in the corner of a small room in the Cavendish Laboratory, just down the road from the Eagle. It's a sculpture made out of what looks like tinker toys with wedges of cardboard attached. The sculpture is hardly impressive. It's something a kindergartner would build. But if you look closely, it has a certain elegance to it, a pleasing symmetry. It's shaped like a long ladder with many rungs. Except it has been twisted so that it looks more like a spiral staircase. This is the first accurate model of DNA, the molecular blueprint for life. DNA's structure is one of the most important discoveries in the history of science, and Francis Crick has just announced it to the world in a pub. The discovery of DNA's double helix would go on to earn these two scientists a Nobel Prize and would transform science forever. But of course, it wasn't their discovery, at least not theirs alone. It had taken decades and the work of many other men and women to get them there. It may have sounded incredibly arrogant at the time, but in one sense, Francis Crick was right. Their discovery would unlock the secret of life. American Innovations is pleased to have ZipRecruiter as its presenting sponsor. ZipRecruiter strives to lead the way in innovative talent recruitment for businesses of all sizes. It's the smartest way to hire. Their powerful technology sends your job post to over 100 of the web's leading job boards. And it doesn't stop there. It learns from your feedback to better understand what kind of candidate you're looking for. It's so effective that 80% of employers who post on ZipRecruiter get a quality candidate through the site within the first day. Stay tuned at the end of this episode for an inside look into ZipRecruiter's culture of innovation. Ryan Eberhard, Senior VP and Head of Product at ZipRecruiter, is back again with his favorite innovation that helps ZipRecruiter constantly improve their technology to connect job seekers and employers at businesses of all sizes. If you love what you're hearing, now you can try ZipRecruiter for free. Just go to ZipRecruiter.com/AI. That's ZipRecruiter.com/AI. ZipRecruiter, the smartest way to hire. From Wondery, this is American Innovations. I'm your host, Stephen Johnson. This is episode 3 of our 6-part series on DNA, The Stuff of Life. In our last episode, we looked at how scientists in the fly room at Columbia University made several major discoveries. One was that genetic information, in the form of genes, is carried around on structures called chromosomes. But what exactly are genes, and what are they made of? Finding the answer to that question would occupy many of the greatest scientific minds of the 1950s. But the story of that search was also a very human affair, full of stupid mistakes and petty jealousies and, as you might have guessed from that display in the Eagle pub, more than a little arrogance. In the next chapter in the story of DNA, 4 figures stand out. James Watson and Francis Crick, obviously, and a third man, Maurice Wilkins. The fourth name is sometimes overlooked, Rosalind Franklin. We'll get to her soon. Watson claimed that he had discovered the secret of life with good reason. But the secret story behind its discovery proves just how messy science, and life itself, can get. By 1950, scientists knew that chromosomes carried genetic information, and they knew that they were made of 2 ingredients, proteins and nucleic acid. The key to inheritance, the secret sauce of genetics, had to be in one of those. And at the time, most scientists were sure that it was the proteins. Now, you can think about genes as a way to store information. And in storing information, cells can use 2 different languages, proteins or DNA, each with different alphabets. A protein is a long chain made up of smaller bits called amino acids. Amino acids are the building blocks of proteins, just like A, B, C, and D, and so on are the building blocks of words. There are 20 amino acids in total, 20 letters. DNA is also a long linear molecule. But instead of amino acids, DNA letters are called bases. There are 4 bases total: adenine, cytosine, guanine, and thymine, abbreviated A, C, G, and T. The key point for scientists is that while proteins have 20 amino acids, 20 letters to work with, DNA has just 4 letters, A, C, G, and T. And if you're trying to write down an instruction book for life, a book with enough power to create ants and redwoods and duck-billed platypuses and human beings, an alphabet with 20 letters seems far more powerful than an alphabet with just 4. Imagine trying to write a novel in a language with just 4 letters and you can see the problem. Naturally, most scientists believed that the bigger alphabet carried genetic information. Until the early 1950s. When 2 scientists with some vats of viruses and a kitchen blender make a dramatic discovery. It's 1952, and in Cold Spring Harbor Laboratory on Long Island, 2 scientists are bent over a wooden workbench. Lining the laboratory are shelves of beakers, flasks of murky liquids with microbes brewing inside. The lead scientist is Alfred Hershey. He's a scrawny, bespectacled man with an army buzz cut and a mustache. The assistant at his side is Martha Chase, a round-cheeked woman with close-cropped brown hair. Ready? I've prepped the bacteria. Hershey and Chase want to prove once and for all that the genetic material being injected isn't DNA, but proteins. To this end, they've designed a clever test. Let's add the viruses now. They take down 2 flasks from their shelf. One flask has a batch of viruses with radioactive DNA, and another has radioactive proteins. Hershey and Chase can now trace the movement of DNA or proteins by following the trail of radioactivity. It's like a radioactive tracking device that's been placed on the DNA and the proteins. They squirt viruses from each batch into 2 separate Petri dishes full of bacteria. They give the viruses time to inject their genetic material into the bacteria. Then they use a kitchen blender and a centrifuge to separate the virus husks from the bacteria. Now comes the moment of truth. Using a Geiger counter, they examine the bacteria infected by the virus with the radioactive proteins. If proteins are the genetic material, then those bacteria should be radioactive as well. But nothing happens. The Geiger counter remains silent. No proteins have been injected. But when they point the Geiger counter towards the other batch of bacteria, it can only mean one thing. The virus has injected not proteins, but DNA. Against all expectations, here is the genetic material. Genes aren't made of proteins at all. They're made of DNA. After Hershey and Chase's blender experiment, DNA becomes the hottest biomolecule in the world. But if the blender experiment answered one question, it raised many others. What is the structure of this DNA stuff? How do cells copy DNA and pass it from parents to children? And most perplexing of all, how can 4 measly letters encode enough information to make every living thing on Earth? The first step to answer any of these questions was to determine the structure of DNA. The starting gun had been fired on a scientific race. The goal? An accurate model of this crucial molecule. And at the time, there was only one man to beat: Linus Pauling. It's the spring of 1945. Linus Pauling attends a medical conference in New York. After a full day of lectures, he shows up for a dinner where yet another doctor is giving another presentation, this time on a painful blood disorder called Sickle-Cell Disease. With this disorder, we'll typically report pain in the joints. Other symptoms include dizziness, nausea. But Pauling isn't paying much attention. It's been a long day and he's pretty tired. Suddenly, though, the doctor says something that snaps Pauling awake. Sickle-cell disease produces misshapen red blood cells. Instead of being round, the cells of people with the disease are crescent-shaped. But that crescent shape is far more common in veins than arteries. Red blood cells and arteries tend to look normal. The health consequences. Pauling has a flash of insight. It would take a few more years to work out the details, but his insight that day would change history. The Wizard of Caltech. That's what people called Linus Pauling, a tall, angular chemist with curly brown hair and a big grin. He was widely considered to be the best chemist in the world. In the 1920s, he had figured out how to apply quantum mechanics to chemistry. Pauling single-handedly revolutionized the field. A colleague said that after Pauling, chemistry could be understood rather than being memorized. Not content to revolutionize just one field, he branched out into biology in the 1940s. People with sickle-cell disease have an incorrect amino acid at one point in their hemoglobin. This mistake changes the structure of hemoglobin so that it doesn't function properly and is bent out of shape. What made Pauling's discovery so important was that the implications went way beyond sickle cell disorder. This was the first disease ever traced down to a molecular flaw, and it opened up a whole new approach to medicine. Doctors could now use biochemistry to study the root causes of diseases, instead of just treating symptoms. Pauling was once again hailed as a genius. So when Pauling began to work on DNA, he was the odds-on favorite to discover its structure first. If anyone could do it, his colleagues figured, it was the Wizard of Caltech. But several thousand miles away, in a pub in Cambridge, England, 2 awkward graduate students were already plotting to beat him to the punch. What if you could pull up a chair next to some of the wisest, most accomplished teachers, creators, changemakers, and leaders in the world, and ask them your deepest questions and learn from their wisdom? Well, that's exactly what award-winning author and innovator Jonathan Fields does on his wildly popular Good Life Project podcast. And he's inviting you to come along. Every week, Good Life Projects bring you raw, unscripted, revealing conversations with luminaries like Brené Brown, Elizabeth Gilbert, Reverend Angel Kyoto Williams, and Tim Ferriss. No script, no sound bites, no standard Q&A or 3-minute segments. These are in-depth, often deeply intimate and surprising conversations. You'll not only see a side of these often high-profile guests that you'll rarely ever see, you'll also hear stories and wisdom that will move you deeply, reconnect you with what it means to be alive, and inspire you to craft your best life. Start listening now. Just search for Good Life Project in your listening app. Figuring out DNA's structure was a real puzzle. It wasn't possible to look at the molecule, at least not in any great detail. So scientists had to deduce its shape based partly on the little they did know about it. For a start, they knew that DNA had 2 main parts. There were its bases, the A, C, G, and T. And they also knew that it had a backbone. The backbone consists of alternating units of ringed carbon and phosphate molecules. The A, C, G, T bases then stick out from this backbone. But beyond that, they didn't have much of an idea of the rest of the molecule's shape. Was the spine straight or twisted or some other shape entirely? Nor did anyone know how many strands of DNA were involved. Was there a single backbone, or were there multiple strands coiled around each other? These were the questions that the two Cambridge graduate students, James Watson and Francis Crick, thought they could solve. In 1951, Crick is 35 years old, ancient for a graduate student. He'd started out in physics and then gotten distracted with war work for England before switching to biology. He's a balding blonde man who dresses in three-piece suits and wears his sideburns long. He also talks nonstop. A colleague once described his voice as, quote, an incessant falsetto with occasional nuggets gleaming in the turbid stream of prattle. He declared that together, Crick and his colleague Watson look like a variety act. James Watson is 10 years younger than Crick, just 25, and has wild, unkempt hair and ears that stick out from his head. He's an American and has actually ended up in Cambridge after failing to get into Caltech, where Linus Pauling worked. Watson had actually been working on those same kinds of viruses that had wound up in the Blender experiments a few years earlier. He's always been interested in the structure of DNA, but in the early 1950s, he attends a lecture in London that seems to spark something inside him. He half listens to the scientist as she outlines some preliminary work she's done on DNA. It involves firing X-rays at a sample of DNA and then capturing the reflected rays on photographic film. From the patterns that show up, she can infer a little about the structure and how the backbone and bases fit together. It's new information that Linus Pauling, all the way on the other side of the world, doesn't yet know. When he gets back to Cambridge, Watson tells Crick about the lecture. We can build a physical model of DNA based on this data. We can solve the problem before anyone else. They get to work immediately. Let's approach it like a logic puzzle. There are some things we know for certain, right? DNA has a backbone rich with phosphorus, and it contains the 4 bases, A, C, G, and T. So we need to figure out how these parts fit together, right? What did Franklin say in the lecture about the shape of DNA? It has a backbone, right, but what else? I'm not sure. They have no choice but to work with the details they have. To build the model, they use what looked like an erector set. Watson once called their tools tinker toys. The DNA backbone is made of short metal rods and rings and is held together with clips. The knobs, or bases, are flat plates of sheet metal or cardboard. They start at the floor and begin building toward the ceiling, clamping everything to a metal pole in the center. One big question is, how many backbones to include in the model? Watson and Crick discuss the matter over gooseberry pie at the local pub, the Eagle. Based on Watson's half-remembered data, they decide on a 3-backboned DNA molecule. There's a lot of cramming to make things fit, but by the next morning, they've slapped together a snappy-looking triple helix. The next thing to do is get some feedback. Crick calls up Maurice Wilkins, the head of the London group studying DNA. Wilkins decides to come to Cambridge the next morning to see what they've come up with. He's accompanied by the X-ray specialist, whose lecture Watson had attended, Rosalind Franklin. Watson and Crick don't realize, however, that things within the London group are tense. There have been fights recently over equipment, over access to DNA, and especially over who was really in charge of their work, Wilkins or Franklin. The trouble had started a year earlier. A Swiss biologist had developed a recipe to isolate very pure DNA from the thymus gland of a calf. It came out as a goopy gel, whitish in color. He kept his recipe a secret, but was very generous about sharing samples of the DNA, and gave a vial to Wilkins at a scientific meeting in May of 1950. Wilkins takes the DNA goo back to his lab, storing it in an old jelly jar. Wilkins then tries using X-rays to photograph the DNA and determine its structure. But being inexperienced, his pictures are mediocre, too blurry to be useful. After several months of frustration, he admits he needs help and he asks his boss, a scientist named J.T. Randall, to find an expert on X-ray photography to assist him. Randall does. Rosalind Franklin. Although she's English, she's been working in Paris for years and she loves the egalitarian atmosphere in the labs there. She doesn't feel like a second-class citizen there. But Franklin ultimately wants to establish herself in England, and so she accepts the job at King's College back in London to work on DNA. And here's where the trouble starts. When Randall hires Franklin, he assures her that she'll be working independently, running her own research project. But this promise contradicts what Wilkins actually wanted. He only wanted an assistant. And unfortunately, Wilkins is on vacation when Franklin accepts and can't clear the matter up. So when Wilkins returns from vacation and starts treating Franklin like the assistant he expects, she bristles. He keeps hounding her for data, and she keeps telling him to back off. One afternoon, things get especially ugly. Franklin has succeeded in producing a much tighter beam of X-rays than Wilkins ever has, and is getting sharp pictures. Say, these are really good. Do you see how these two lines cross here? That seems to suggest a helix shape. How dare you interpret my data for me? Franklin snaps. Wilkins is taken aback. All I meant was maybe we can collaborate. I don't think that would be appropriate. After that, the atmosphere in the London lab is poisonous. By the time they get the call from Watson and Crick, Franklin and Wilkins are barely speaking to each other. For their part, Watson and Crick are oblivious to this drama. They're too excited about their triple helix. They start off their presentation enthusiastically. Thank you both for making the trip at short notice. As I already mentioned, we've developed a theoretical model for the structure. But things quickly unravel. As theorists, not experimentalists, the duo has very little practical knowledge about DNA, like the kind of knowledge Franklin has. And that morning, she uses that knowledge to trash their model. The conclusion that Rosalind's X-ray data suggests a helix. It's much too early to conclude that. Can I also point out an immediate problem that comes to mind with your model? Are you too familiar with the research? Franklin is arguably the better scientist, and she knows the Cambridge duo's model can't be right. It's a humiliating ordeal for the two men. Even the blabbermouth Crick falls quiet. Their lunch at the Eagle that day is a gloomy one. Worse still, word of Franklin's dressing down soon trickles up to their advisor at Cambridge. Technically, both Watson and Crick are supposed to be working on other projects to earn their PhDs. The advisor has tolerated the DNA work as a sideline, nothing more. But after Franklin and Wilkins leave, he calls them into his office and in no uncertain terms bans them from working on DNA. Watson and Crick swallow hard and reluctantly agree. But neither man has any intention of keeping this promise. If they have to work on DNA in secret, so be it. Meanwhile, Linus Pauling is dealing with troubles of his own. The U.S. government is accusing him of treason. The problem isn't his research, it's his political activism. In particular, his speeches and articles opposing nuclear weapons. It doesn't take long for people to start whispering that Pauling must be a communist himself. So in early 1952, Pauling applies for a passport to travel to Europe that spring. The State Department denies it. The department is of the opinion that your proposed travel would not be in the best interests of the United States. Pauling fights back, but as a result of all this, he misses an important biochemical conference in England on May 1st. Rosalind Franklin does attend the conference and talks to one of Pauling's assistants there. She even shows him some top-notch photographs she's taken of DNA. But Pauling himself would never see the pictures. Eventually, after Albert Einstein and other scientists raise a stink, Pauling does receive a limited passport to travel to Europe in June of 1952. And before the trip, he writes Maurice Wilkins. I'm traveling to London. I'm very interested in seeing Rosalind Franklin's photographs during my visit. I'm sorry, but since the conference, we've decided not to share the pictures anymore. The window of opportunity has closed, and Pauling's passport troubles have cost him his one and only chance. As a result, Pauling is at a major disadvantage when he finally sits down in the fall of 1952 to crack the mystery of DNA's structure. Not having seen Franklin's pictures, he has to rely on a few sketchy photographs taken by another colleague. And unfortunately for him, the DNA in these pictures is partially dehydrated. That's a problem because dehydrated DNA shrivels up and changes shape. It would be like trying to reconstruct what a flower looks like, based solely on a dried-up bouquet, or more precisely, a poorly lit picture of a dried-up bouquet. But Pauling doesn't know this when he starts. The structure he comes up with has 3 backbones, and the bases are pointing outwards. Pauling then rushes the paper into print, submitting it to a journal on the last day of December in 1952. The Wizard of Caltech has done it. He's described the structure of DNA before anyone else. Or so he thinks. It's January 1953. Peter Pauling, Linus Pauling's son, has just returned to university after Christmas holidays. He's at Cambridge University, the same university as James Watson and Francis Crick. James, Francis, how were your holidays? Never mind about that. Have you heard anything about your father's work on DNA? Peter has. Pauling has written a paper on DNA, he tells Watson, and will be sending him a copy shortly. Watson's stomach clenches. He's convinced the Wizard of Caltech has pulled some DNA rabbit out of his hat and beaten them. He distracts himself by actually working on his PhD research for once. But try as he might, those 3 letters keep pounding in his head. DNA. DNA. Finally, one day in early February, right after lunch, Peter strolls into Watson and Crick's office with the paper in his pocket. As soon as Peter walks in, Watson sees something in his expression. Watson's stomach drops. Taking pity on them, Peter cuts to the chase. I've read it. He's come up with a model of DNA. It's a triple helix with the backbone facing the center. What's wrong? That was our idea. We came up with it a year ago, but we got told that it just couldn't work. If only we had stuck with it, we could have solved it already. Unable to contain himself, Watson yanks the paper out of Peter's coat pocket and begins skimming. Oddly, though, he notices some errors near the beginning. He says that the backbone has to face the center, but Franklin told us that was impossible. Then more errors pop up. Crick is reading over Watson's shoulder by now and points out other flaws. And look, here, we said exactly the same thing, but that can't be right. Finally, it dawns on the 2 men. The great Linus Pauling has just reproduced, in a soon-to-be-published paper, every rookie mistake that they had made a year earlier. Realizing they haven't lost the race yet, they run to their advisor and confess that they've been working on DNA this whole time. In high spirits, Watson and Crick storm into the Eagle that night. Rather than his traditional glass of sherry, Watson lets Crick buy him a whiskey to toast Pauling's blunders. The race for DNA isn't over, provided Watson and Crick can wrestle a few clues away from Rosalind Franklin. It's February 1953. A young woman is bending over a light box at King's College in London. She has short, wavy brown hair and is intensely focused on the photograph in front of her, a picture of the most talked-about molecule in science, DNA. As one of the few female scientists at the college, in fact, in all of England, Rosalind Franklin has had a tough time at King's College. She misses her lab in France, where she'd been treated with more respect. Here, she's barred from entering some of the common rooms, which are male only. She can't even eat her meals in the same places as the men either. All that hassle takes time away from the work she wants to be doing on DNA. She sighs, straightens up, switches off the light box. Just then, the door bursts open, and a skinny young man with wild hair barges in. Her eyes narrow when she sees who the intruder is, and she picks up the photo and tucks it away in a drawer. You might have knocked. Oh, hello, Rosie. Still wearing the same clothes as last time I saw you. Some things don't change, I suppose. Such a shame. You know, if you put on a little lipstick, maybe a new dress. What do you want? James Watson saunters up to the bench. Then he removes a paper from his pocket. Peter sent me a copy of his father's article. Want to look? He's spoiling for a fight. It might amuse you. It's essentially the same structure Francis and I suggested last year. Remember, the one you so graciously tore apart? Anyway, get this. He still thinks it's a triple helix. But there's not a shred of evidence for that. Doesn't anyone around here care about doing proper scientific research? Watson rolls his eyes. we both know the simplest form is a helix. If you weren't so incompetent at interpreting your pictures, you'd see that. Franklin stands up, angry. She starts coming around the lab bench towards him. Watson starts backing up towards the door, his confidence deserting him. Just as Franklin is about to reach him, the door swings open again. "James, are you here?" A skinny, bespectacled man with a huge forehead is there. He's about to say more, but senses the tension in the air. He glances at Franklin and then back to Watson. I heard you were looking for me." Watson ducks behind him. Yes, I was just leaving. Miss Franklin decided our conversation was over." A near fight like this really did take place in 1953. Only the timely arrival of Wilkins saved Watson's neck. And here's where the story of DNA takes a controversial turn. By nature, Wilkins is timid and reserved, but as he and Watson scurry down the hallway, Wilkins opens up about the emotional hell he says Franklin is putting him through. In fact, Wilkins is so relieved to talk to someone that he does Watson a favor, a potentially unethical one. During their conversation, Wilkins mentions that Franklin took some beautiful pictures of DNA a few months ago, pictures far clearer than any previous ones. But for some reason, she's hidden them away, then gotten distracted on a side project. Watson, naturally, asks what the photographs look like. At this, Wilkins leaves the room and comes back with a copy of one, a copy he'd taken earlier without Franklin's knowledge. It's now known as Photograph 51. He then hands it to Watson, something Franklin never would have allowed. To an amateur eye, Photograph 51 doesn't look like much. A black X on a white background, reminiscent of a wristwatch. But to someone trained to interpret these photographs, it's stunning. "The instant I saw the picture," Watson later said, "my mouth fell open and my pulse began to race. Franklin has been insisting for months that they don't have enough evidence to conclude DNA is a helix. But to Watson, this black cross can't be anything but a helix." Watson races back to Cambridge to tell Crick, and over the next few days, the clues begin to fall into place. Clue number 1 is Photograph 51. Based on that, they feel certain there's a helix involved. Clue 2 had come during Franklin's original dressing down a year earlier. The bases on the backbone have to be on the inside of the molecule. This was where they had gotten stuck before. How do those bases match up when they're facing inwards? Watson and Crick realized that perhaps their initial idea about there being 3 backbones to the molecule was wrong. What if DNA isn't made up of 3 corkscrew shapes twisted around each other, with the bases crisscrossing each other? What if it was just 2 corkscrews, a twisted ladder shape, a double helix? And when they look at the shape of A, C, G, and T more closely, their hearts begin racing again. Watson and Crick's model finally comes together one morning in late February. That very day at lunchtime, as Watson later recalled, Francis winged into the Eagle to tell everyone within hearing distance that we had found the secret of life. It's a great moment in the history of science. But the celebration actually leaves Watson queasy. He's worried they're being premature. After all, they still haven't nailed down all the details, much less published their results. Who knew what tricks the wizard of Caltech might yet pull? The two press on, rushing to hammer everything out, and in April of 1953, they published their findings in the journal Nature. But why exactly is the double helix so famous? What's the big deal? For one thing, it was an elegant solution. In fact, it was the simple beauty of the 2 twisting corkscrews that convinced Watson and Crick that they were on the right track. But most importantly, DNA's shape told Watson and Crick something vital about how it works. Its form was its function. To understand why that is, you first need to know something else about the Tinkertoy model they came up with. When you look closely, you can see that the bases, remember, those are the rungs of the ladder, A, C, T, and G, always come in pairs. They fit together like puzzle pieces, connecting two sides of the ladder. A always goes with T. C goes with G. When DNA replicates, it first splits down the middle. A and T, C and G, they're separated. Basically, the ladder gets split in half. But here's the key part. Because those bases always come in pairs, no matter which side of the ladder you have, the cells are able to figure out what the other half should be. A's go with T's. C's go with G's. And then the same with the other side of the ladder. In the end, they've built two exact copies of the original ladder. Two strands of genetic code. That's the reason Watson and Crick are so famous. Not just for discovering the shape of DNA, but because they realized that the shape revealed how DNA worked. As they said in their initial paper on DNA, with cheeky understatement, it has not escaped our notice that the specific base pairing we have postulated immediately suggests a copying mechanism for the genetic material. In the spring of 1953, Linus Pauling catches wind that the Watson and Crick Variety Act over in England might have scooped him. He happens to be traveling to a conference in Belgium in April and immediately schedules an emergency trip to Cambridge. Watson and Crick must have been nervous as the great wizard of Caltech entered their office that Saturday morning and cast a critical eye over their twisting double corkscrew model. But to Pauling's credit, he handles the situation with grace, quickly conceding that they've beaten him. Losing like this stings him. He's a proud man and a good scientist. But the double helix is so elegant and makes sense of so much data that he knows they're right. Pauling even helps promote Watson and Crick's discovery over the next few months, giving their careers a huge boost. Unfortunately, Watson and Crick's relationship with the other characters in this story prove less collegial. Things get downright prickly, in fact. Watson and Crick do cite Rosalind Franklin in their paper announcing the double helix, but only briefly. Franklin and Maurice Wilkins also published their own paper on DNA in the same issue of the same journal. But for whatever reason, probably a combination of Wilkins' retiring personality, Franklin's gender, and the fact that Watson and Crick discovered how DNA copies itself, the King's College crew fades from the world stage. To add insult to injury, Watson publishes a book called The Double Helix in the 1960s that paints a nasty portrait of Franklin. No one ever claimed that she was easy to work with, but the book makes her look terrible. He insists on referring to her as Rosie throughout, a nickname she hated, and he makes disparaging comments about her clothes and lack of makeup. It's July 2017. It's morning, and the Eagle Pub in Cambridge is not open yet. Back in 2003, a blue plaque was placed on the outside of the building to commemorate its part in the discovery of DNA. It says, "It was here on February 28, 1953, that Francis Crick and James Watson first announced their discovery of how DNA carries genetic information." Today, though, there's something else on the plaque. A passing Cambridge University linguist is one of the first to spot it and takes a picture. Someone has either defaced or, depending on your point of view, corrected the plaque. Next to Watson and Crick's name, they've written two words in thick black ink across the bottom: plus Franklin. The story of the double helix is an epic one, full of conflict, drama, and deceit. Most people don't realize, however, that solving DNA was just a prelude to another, and in some ways bigger, story. We had finally nailed down what DNA looked like and how it worked. Now we had to figure out all that DNA does. How does it build creatures? What does each gene do? Luckily, the next grand chapter in the saga of DNA, the Human Genome Project, was just around the corner. And it would prove no less glorious, no less controversial, and no less messy. At the beginning of this episode, we introduced you to Ryan Eberhard. He sat down with Leah Sutherland, a producer on this show.
My name is Ryan Eberhard. I'm the SVP and head of product at ZipRecruiter. Hey, Leah, how are you?
I'm doing pretty well. How are you?
I'm great.
Did you have a favorite science class in school?
My favorite class in university was actually Introduction to Physics. It was taught by a legendary physics professor who wrote our textbook. His name was Hugh Young, and he opened every lecture with a Shakespeare quote. And he was just this incredibly dynamic man with an infectious love for the study of physics.
Do you think it was his energy and his love for it that made you enjoy it?
I think it was. Yeah, I think it was. It's often thought falsely that the study of mathematics and science is passionless and rote, but actually some of the most passionate people I've met have been from the sciences.
There is a nice kind of like artistry and philosophy behind science and mathematics that I think gets looked over a lot.
There is. That's why musicians so often love mathematics.
Actually, in future episodes of American Innovations, we turn genetic and mathematical concepts into music, so listen up for that. You can learn more right now on how ZipRecruiter is changing the game. Their technology learns from your feedback to better understand what kind of candidate you're looking for. It's so effective that 80% of employers who post on ZipRecruiter get a quality candidate through the site within the first day.
And now our listeners can try it for free. That's right, free. Just go to ZipRecruiter.com/AI. That's ZipRecruiter.com/AI. One more time, ZipRecruiter.com/AI. ZipRecruiter, the smartest way to hire. I hope you enjoyed this episode of American Innovations. If you did, please do give us a 5-star review and tell your friends to subscribe. We're available on Apple Podcasts, Stitcher, NPR One, and every major listening app, as well as at Wondery.com. If you love the info you heard, the author of this series, Sam Keen, has several wonderful books about DNA, neuroscience, and more. His book on DNA is called The Violinist's Thumb, and it's available wherever books or ebooks are sold. You can learn more about his books on his website, samkeen.com, or you can follow the links in the show notes. And a quick note about these historical recreations you've been hearing. In most cases, we can't know exactly what was said, but these scenes are based on real historical research. If you're listening on a smartphone, tap or swipe over the cover art of this podcast. You'll find the episode notes, including some details you may have missed. You'll also find some offers from our sponsors. Please support our show by supporting them. And thank you. American Innovations is hosted by me, Stephen Johnson. For more information on my books on innovation, like How We Got to Now and Wonderland, you can visit my website at stephenberlinjohnson.com. Sound design on this episode is by Bay Area Sound. This episode is written by Sam Keen. The producer is George Lavender. Executive produced by Marshall Louis for Wondery.